Hey everyone, today this is just a quick video to go over DNA replication and how to tell which direction the leading and lagging strands are moving in. So if we remember DNA replication, DNA is a double-stranded molecule like this, and replication starts at here what we call an origin of replication, where a helicase and a bunch of other helper molecules come to unzip the DNA in both directions. So simultaneously, it's replicating this way and this way. And in one direction of each strand, there's a leading strand, a continuous strand, and the other direction is a discontinuous strand due to the nature of the organic chemistry behind it, which we're not going to talk about. So to identify which way the leading and lagging strands are going, first you start at the center. And notice how there are two strands here. Each strand gets copied at the same time. So first we're going to put down a primer. That's the first thing we start with. And the direction of the primer has to be opposite with respect to its template strand. So for this primer, this top strand is its template strand. For the bottom primer, this bottom strand is the template strand. So when we're talking about directionality, we're talking about it with respect to its template strand. So since this end is the 5' end, this end of the primer has to be 3'. And since this end of the template strand is 3', this end has to be 5'. And on the bottom strand, it's the same idea, but opposite. So this is the 3' end, so this has to be 5' on the primer. This end is 5', this end has to be 3' on our primer. So far, so good. So now we have to determine which direction we move. Notice here, a key point about DNA is that we move from 3' to 5', but we build from 5' to 3'. Now that sounds confusing, but to do this, to determine which way we move, you focus on the 5' end of the primer. And you draw the arrow going from 5 to 3 like this. This 5 to this 3, and it keeps going like that. But notice that as our new strand is growing from 5 to 3, with respect to our template strand up top, we're actually moving from 3 to 5. And the same scenario happens down here. We focus on this 5' of our new strand, and we go from 5 to 3 in this direction. These two strands are our leading strand. And for the other two halves of the strands is the lagging strand, where we actually have to put down multiple primers to replicate those. So we put down another primer, let's say here, this is 3', this is 5'. But notice we can't go from 3' to 5' like this. The chemistry just doesn't work that way. We have to go this way. Which means every time the helicase continues to unzip this, we have to put down a new primer in the same direction and just keep going this way. So this here would be a lagging strand. And up here, it's the same idea. A primer going from 3 to 5. And we always have to move from 5 to 3. Helicase opens up the bubble a little more. And once again, we go 5 to 3. So this is also a lagging strand. So notice there's 4 things happening at the same time. And that's how you determine direction of leading and lagging.